Hello everyone, my name is Levy and I will present to you the work that is entitled Impact of Point Variations in HLA-DRB1 on the Stability of pHLA complexes and Susceptibility to Multiple Sclerosis. This investigation arose from the need to better understand the molecular mechanisms that link specifically HLA-DRB1 variants to the risk of developing this disease. So far, the exact causes of multiple sclerosis are not fully comprehended yet, but we know that both genetic and environmental factors are involved. From a genetic perspective, most of the genes associated with multiple sclerosis are located in the major histocompatibility complex, with the DRB1 1501 allele standing out, often referred to as the multiple sclerosis gene. In addition, many environmental factors, such as vitamin D levels, sun exposure, infections, obesity, and smoking, also play important roles in the development of this disease. Among these factors, the Epstein-Barr virus, EBV, is particularly notable as it has been shown that almost all multiple sclerosis patients had prior contact with EBV. It is believed that viral antigens for EBV, such as one of its proteins, EBNA1, may act as triggers for multiple sclerosis. Focused on genetic factors, our research group is investigating HLA-DRB1 alleles, a high polymorphic protein. We are studying how different alleles can influence susceptibility or resistance to multiple sclerosis. For example, the alleles 1501 and 1503 differ only at position 30 in the primary sequence, yet both share the same peptide interaction motif and have the ability to present this MBP peptide, which is known as an important autoantigen in the pathogenesis of multiple sclerosis. In contrast, the allele DRB1 1502 differs from 1501 only at position 86 and shows no association with multiple sclerosis. Despite the abundance of biochemical and structural information available for these alleles, it is still unclear why some confer a higher risk for this disease, while others seem to offer protection. We know that polymorphic residues in the binding groove of HLA-DRB1 are crucial for peptide binding and recognition by the T-cell receptor. Changes in this residue alter the nature of binding pockets in HLA class II, where the peptide side chains fit, which may be important for understanding the different immune response associated with each allele. To evaluate the effect of these point mutations, molecular dynamics simulations were performed on predisposing and non-predisposing alleles, both unbound and in complex with relevant peptides. The study methods are summarized in this flowchart. To perform the MD simulations, the complexes underwent several preliminary steps. First, the crystallography structure was obtained, followed by the filling and correction of missing residues using comparative modeling. Next, polymorphisms associated with multiple sclerosis, either as risk or protective factors, were modeled. The peptides were then subjected to docking. And finally, the system was prepared for the MD simulations, aimed at capturing the dynamic profile of molecules based on Newtonian mechanics principles. From there, the pathways were generated and evaluated using MD methods, such as RMSD, RMSF, and others. Regarding the results, the RMSD values demonstrated structural stability in all complexes. However, the behavior of the complexes indicates that the binding of the peptide, or its absence, did not result in major change in the global structure of the HLA-DRB1 alleles studied. This figure shows that fluctuations were observed exclusively in alleles associated with susceptibility. The segment encompassing residue 61 to 80 in the DRB chain exhibited greater fluctuations in susceptibility alleles, when bound to MBP 8599, while the segments from residues 48 to 52 showed high fluctuation peaks in the DRB1 1501 when bound to EBNA1. Additionally, the complexes with EBNA1 displayed a greater average distance between the master centers of interaction groove helices, indicating an opener conformation compared to the complex with MBP and CLIP. The RMS results calculated for the peptides showed that MBP remained stable in the interaction groove of allylic isoforms, whereas the N-terminal of EBNA1 exhibited greater fluctuations in 1502. As additional information in this presentation, this table presents the MM-PBSA binding free energies for the alleles, showing that the MBP peptide exhibits a smaller difference in interaction energy with the protective allele. In conclusion, these findings suggest that allelic variations of HLA-DRB1 within the DR15 group differentially affect the stability and dynamics of pHLA complexes, potentially influencing susceptibility to multiple sclerosis. These insights may deepen the understanding of multiple sclerosis genetic bases and, more importantly, offer valuable guidance for developing more targeted therapies. The next steps in this study involve ongoing investigations into the interactions of HLA-DRB1 with peptides and TCRs involved in multiple sclerosis, aiming to elucidate the roles of specific residues in multiple sclerosis susceptibility. Concluding this presentation, we would like to thank CNPq, CAPS, FAPEST, the Graduate Program in Genetics, FAEPA, the National Laboratory for Scientific Computing, and the Santos Dumont Supercomputer for the support and computational resources that made this work possible. We are available to answer any questions via the provided emails. Thank you very much.